the path from Abelson leukemia virus to Gleevec. The main objective of this lecture is to take you to the decisions scientists need to make when they try to develop drugs. What is the process? What are the conceptual problems scientists face that lead them to take go or no-go decision? And I will do it in a high resolution with quite a bit of personal point of view. I will also share with you the most important take-home lessons I personally have from the discovery of Gleevec. The other personal things, who did what, is not the most important things in this lecture. However, as this is an integral part of the story, you will hear about them as well. Discovery of drugs. Why we are making drugs and what are the steps that we need to take. First of all, you need to determine an unmet medical need. You have a disease and you don't have a good answer how to treat the disease. That's the first step. And then, when it comes to conventional drugs, the drugs is likely to do something specific which is related to the mechanism of action of the disease. If this is an enzyme which is causing the disease, sometimes inhibitors for these enzymes can work. So you need to find the mechanism of action of the disease and to find what are the genes and the protein products that are involved with the disease. In general, you want to identify a target protein because drugs are going to work on proteins. Inhibitors of enzymes or changing the activity of the protein in some other ways. So you need to understand the disease. You need to find the most important players of the disease. And then to ask a question, is the target druggable? That's what we call proof of concept. And here we ask some conceptual questions. Can we act against the target protein? And in what strategy? Most of the drugs that we have on the shelf are competitive inhibitors that are working on certain enzymes. This is one way to deal with the target protein. And second, can we make drug which is going to be selective? From conceptual point of view, we don't want the drug to cause damage more than good. So the strategy that we need to use is a strategy that will give a benefit more than harm. If in all those stages that we had so far, we are going to have the go decision, in other words, that we have a positive result, we are moving further towards screening many compounds against the target proteins. And when you have some good results in this stage, you take the drug to the chemistry lab to make derivatives. When you have a certain lead that can give you a certain activity, usually it's not going to be the full activity immediately during the screening, but a lot of time you can take the promising leads to the lab in order to make some derivatives and then to concentrate on few of those which are working at the best. And from here you start to do some more tests on animal models, starting with mice, and then we can go to larger animals in order to check the toxicity of the drug. And second, to see if the drug can reach the appropriate place. If the problem is in the brain, you need the drug to cross the blood-brain barrier. Toxicity, for example, can be related to the drugs affecting in a non-specific way the liver or kidneys. And when everything is positive, we move to clinical trials in people. Here we check again for toxicity. This is phase 1. And then we keep checking toxicity for the other parts of the trial, but we also test the efficacy of the drug. So let's go to drug design for chronic myelogenous leukemia, CML. So first, determine an unmet medical need. We have a disease without a cure. People are diagnosed during the chronic phase in most of the cases, still have some 4 to 6 years on average with low level of symptoms. But this is a time bomb, because everybody that was not treated was coming with advanced stages, which is the accelerated phase, and blast crisis, which is a terminal phase. So when you see a person in a chronic phase, the person is quite healthy at this stage, but there is almost nothing that can be done in order to eliminate the problem entirely. One of the treatments that can result with cure is bone marrow transplantation. But bone marrow transplantation is a treatment with a lot of problems. If you are looking here 1 year after the bone marrow transplantation, you can see that in the best scenario, you're losing like 45% of the treated people, when normally without treatment they are going to live for 5 years. When you are doing the bone marrow transplantation later on, 60%, 80% of the treated people are going to die 1 year after the treatment. In many cases, it's a result of the treatment which can accelerate the death of the patient. That's not a good treatment for chronic myelogenous leukemia. But that's what we had during the 20th century. So, determine unmet medical need, checked. There is something that needs to be done. And there is another thing that was quite clear. We knew the mechanism of action of CML. We knew about the chromosomal translocation that created the Philadelphia chromosomes from the 70s. And we were able to find that BCR-ABL is encoded from the Philadelphia chromosomes early 80s. So we had quite a good idea what is the mechanism of the disease and what is the protein which is made and causing so many problems. So during the 80s, we knew quite well that P210BCR-ABL has a clear causal relationship with initiation and the progression of CML. CML is Philadelphia chromosome, and Philadelphia chromosome is P210BCR-ABL. In other words, Philadelphia chromosomes appear in the large majority of CML patients from the initial diagnostic stage of the leukemia. So, if you find a person with Philadelphia chromosome, this person has CML. And if you find a person which diagnosed with CML by other means, this person have the Philadelphia chromosome which is expressing the P210BCR-ABL. The connection of the P210BCR-ABL with leukemia was quite striking. Again, later on during the 90s, we were able to see that transgenic mice which are expressing the P210 BCR-ABL are coming with leukemia, which is quite similar to CML. So that's another way to prove the point that P210 BCR-ABL has a clear causal relationship with the initiation of CML. But there is another data that suggests that Philadelphia chromosome have a causal relationship with the progression of CML, because the appearance of the second Philadelphia chromosome is an indication for the progression of the disease to the accelerated and blast crisis stage. So here we have a clear target for a drug. Proof of concept. The proof of concept here is quite straightforward. Can we act against the target protein? And in what strategy? In other words, the proof of concept was, does the understanding of the genetic abnormality of CML may allow to deliver genotype-specific therapy? And the answer for this question is yes. In May 1996, Brian Drucker and others published their first paper about Gleevec. At that time, STI 571. That's the first description of Gleevec as an inhibitor of the BCR-ABL tyrosine kinase. Drucker et al. Stated quite clearly in this paper that the discovery of Gleevec is a result of an extension of studies by Anafi et al. Those studies happen to be my PhD work under the supervision of Alex Levitsky and Inon Ben-Aria in the Hebrew University of Jerusalem and Hadassah Medical School. I did my PhD work in between the years 1986 and 1992. And the topic of the PhD work was inhibition of the ABL tyrosine kinase activity. On 1986, I started my PhD work with Inon Ben-Aria. At that time, Inon just arrived from David Baltimore's lab, where he gave the final proof that P210 BCR-ABL can cause CML. A year earlier, Alex Levitsky started to work on family of tyrosine kinase blockers, which he designated tyrofostins. At that time, the field of tyrosine kinase blockers was just starting to emerge. A few known tyrosine kinase blockers found in toxic mushrooms like herbimycin and herbstatin. In both cases, the toxic mushrooms gain the toxicity from those compounds, which happen to be very toxic and non-specific. And the people that were working on mushrooms at that time were the first one to figure out that there are certain compounds that the mushroom can make that can inhibit tyrosine kinases. But those compounds were extremely toxic and you cannot use them as a possible drug. But what Alex Levitsky decided to do is to take the structure of herbstatin, and with some imagination, I must admit, he realized that the herbstatin looked like tyrosine, and he suggested that herbstatin must be a competitive inhibitor of tyrosine kinases. And the next step is to make a lot of herbstatin-like molecules the tyrosines, and to find among those type of derivatives some tyrosine kinase blockers that are going to affect a specific tyrosine kinase and not others, so they are going to lose their toxicity. Alex Levitsky's approach to make thousands of tyrosines and to try to fit specific inhibitor to specific enzyme was quite unique at the time. In 1985, I didn't know Alex Levitsky so well, but during my PhD, the first year when I was struggling what will be the topic, the real topic that I will use on the BCR-ABL that I'm working on, I found on my desk an article, a preprint, that Inon left me, and this article was from Alex Levitsky lab. It was talking about the tyrosine kinase blockers that can inhibit the EGF receptor. It took me maybe 5 minutes or so to be in Inon's office and to say, that's it. That's what I want to do for the BCR-ABL that I'm working on my PhD work with the BCR-ABL. Inon drove me to Alex Levitsky lab, and then we realized that we have a lot of things to say to each other. We decided that Alex Levitsky will be the co-supervisor of my PhD work, and I found myself working on this topic on inhibition of the ABL tyrosine kinase activity. Let's talk about biochemistry. Protein kinases have two substrate binding sites. They need to recognize the ATP, so they must have an ATP binding site. The ATP is the phosphate donor. And the second site is the protein binding site. The protein is the phosphate acceptor. So the general idea of tyrosine kinases is to bind the two substrates, the ATP and the tyrosine containing protein substrate, and to put the tyrosine in such orientation towards the ATP so the gamma phosphate from the ATP is going to be transferred to the hydroxyl group of the side chain of the tyrosine. So how you can inhibit this reaction? You can make competitive inhibitors for the ATP binding site. Something that can bind to the ATP binding site, and when the ATP binding site is occupied, the ATP won't be able to bind there. That's a classical competitive inhibitors for ATP. At that time, we had quite a lot of experience to inhibit small molecules from binding to proteins by small molecules. And from the general experience that we had at that time, that was the best scenario. If you can inhibit the small molecule of the ATP with a small inhibitor, that's a fair match. The small inhibitor can stop the binding of the ATP to the same site. So that was one direction. The other direction is to go after the protein binding site, to make something that can bind to the protein binding site, and then when the giant protein is going to approach there, it won't be able to bind. Initially, that seems to be a problem. But when you have such a large protein, the idea that tiny molecule can block the binding of this giant to a protein was not looked so great. At that time, we didn't have a lot of experience in blocking the interaction between large proteins and enzymes. Let's try to understand this a bit further. The interaction between enzyme and substrate it's like a lock and key in some ways. The interaction must be very specific. If you want, you can say the key is the substrate, the lock is the enzyme, and the substrate need to enter into the enzyme and to be modified. So when the substrate have a full productive interaction with the enzyme is going to do its job and to modify the substrate in this way or another, and then the substrate is released after being modified. Competitive inhibitors can be described using this model as cutting a small portion of the key, and now when you put this small portion of the key in the enzyme instead of substrate, in this case the substrate is not going to enter into the enzyme that have the inhibitors inside. The inhibitors going to bind to the same site like the substrate, they cannot bind there together. So the substrate won't be able to fit appropriately, and the enzymatic activity is not going to take place, and the substrate is not going to be modified. No enzymatic activity in this case. Let's go back to the inhibitors for the ATP binding site. What are the meanings of inhibiting the ATP from binding to an enzyme? Well, ATP is a small molecule, and the idea is that there is not too many options for a small molecule to bind to the binding site. So ATP binding site is ATP binding site. But the problem is that 30% of the enzymes that we have are using ATP. And those are essential enzymes. We have a lot of enzymes, normal enzymes that we need for our life that need to work with ATP. Well, you have also the enzyme that can cause cancer, which is using ATP. But if you are going to give the patient with cancer a competitive inhibitor for the ATP binding site, it is likely to affect all the other enzymes as well. And the result here is toxicity, cell death. The patient is going to die. From conceptual point of view, using inhibitors for the ATP binding site seems not to be the appropriate approach, obviously. You don't want to kill the patient, and the idea of making those type of inhibitors seems to be quite problematic, and the scientific community was talking clear and loud against this approach. Targeting kinases was not a popular approach in 1991. It was definitely not a popular approach in 1985 when Alex Levitsky started to work on tyrosine kinase blockers, and it was definitely not a popular approach when I was working on inhibiting the tyrosine kinase activity of the BCR-ABL. As most scientists did not view them as druggable, since it was thought to be impossible to achieve specificity. Makes sense. Scientists were talking very, very loud and clearly. The approach was considered foolhardy. In the dictionary, I found that foolhardy is to describe someone who rushes into action without considering the consequences. And the consequences here can be quite significant. When you develop a treatment with a lot of risk, and there are no other treatments which are available, sometimes this treatment with a lot of risk is going to be used. See the bone marrow transplantation, for example. Bone marrow transplantation is not a perfect treatment. It's causing harm in many cases, more than good. But that's what we have, at least at that time. So the idea was that if you're trying to target tyrosine kinases, you are not going to get a treatment which have low toxicity. You're going to enter into one of those treatments that are highly toxic, but we are still using it in some situations without really getting too far with the benefits that we can have. And I can tell you that in conference that I was there listening to Alex giving a presentation on this topic, the level of the discussion was very loose. Scientists were talking, and foolhardy was not the most severe word that was used against this project, up to the level that another student that was working on tyrosine kinase inhibition of the EGF receptor called me at 2 o'clock at night after a conference like that the words were very, very loose at that time, and asking Moti, what are we doing there? Let's run away. Everybody think that we are crazy. So, yeah. Many years went since this time, but Eric Lander still remember those years. And in this lecture that he gave to students in the MIT, he's talking about it.
There happen to be hundreds of kinases in the cell. Hundreds of kinases. And of course, they all bind ATP in a certain way. And it was pretty clear to people that creating a kinase inhibitor was probably impossible, because all these enzymes had sort of similar binding pockets, and kinases were undruggable. I heard talks about how kinases were undruggable, and good reasons why kinases were undruggable, because they had similar binding pockets and all that. It turned out to be totally false. It turned out, despite the good arguments why it might be impossible to make kinase inhibitors, folks made kinase inhibitors. In fact, the first kinase inhibitors were made against this BCR-ABL fusion protein.
Years later, Brian Drucker was talking about the design of STI 571. Since all protein kinases use ATP as a phosphate donor, and as there are high degree of conservation among kinase domains, particularly in the ATP binding site, it was thought that inhibitors of ATP binding would lack a sufficient target specificity to be clinically useful. However, in 1988, Yaish et al. Reported the first synthetic tyrosine kinase blockers known as tyrofostins, which demonstrated specificity among different tyrosine kinases. The article that Yaish et al. Published in 1988 was about inhibition of the EGF receptor. Pnina Yaish was able to show that certain inhibitors that can inhibit the EGF receptor are not inhibiting the PDGF receptor. And that was the way for them to claim for specificity for those inhibitors. That was the first paper that was around and definitely have quite a lot of effect. Now, in this article, Alex Levitsky decided not to touch the ATP binding site. The ATP binding site seems to be too risky and not relevant for inhibition. What Alex Levitsky did, he started to make derivatives to the epstatin in order to inhibit the protein binding site. So he started to make quite a few inhibitors that can bind to the protein binding site. They look like tyrosine in some ways. By doing so, he was able to inhibit the substrate of the EGF receptor from being phosphorylated. So Alex Levitsky was concentrating in the protein binding site in order to make sure to achieve some specificity, assuming that different substrates are binding to different tyrosine kinases. Here he assumed that some specificity is more likely to take place. And this is from the first article. This is the tyrosine containing peptide. This is the side chain of the tyrosine. And here you can see several family of inhibitors that have some similarity to this region. So the tyrosine project was developed under the assumption that the tyrosine containing peptide has higher potential to be more specific substrate for each tyrosine kinase in order to increase the chance for inhibition selectivity. The tyrosine have been designed to be similar to the tyrosine containing peptide. As you can see, the tyrosines are similar to the tyrosine region of such a peptide. Practically, we were trying to cheat the enzyme and to provide it, instead of its normal substrate, with something else that looked like the substrate. So that was the study that was done by Nina Yaish, Alex Levitsky, and others. And here the main specificity was in between the EGF receptor and the PDGF receptor. Well, why Alex chose the EGF receptor versus the PDGF receptor? Well, both of them are tyrosine kinases, so if you can see differences in between them, you can have your point that the inhibitors can inhibit in a selective way one kinase and not the other. So why Alex chose the PDGF receptor versus the EGF receptor? Well, they're both tyrosine kinases, but they are very different from each other. As you can see here, on the family tree of tyrosine kinases, which is based on multi-sequence alignment, they are quite distinct tyrosine kinases, and you are expecting to have some selectivity in between them, although they are both tyrosine kinases. So selectivity between receptors which are different and have definitely different substrate is one thing, but my problem was totally different. I had to distinguish in between the normal Abel and P210 BCR Abel. From the beginning, it seems like that P140C Abel is an important protein. You don't want to mess up with this one. You don't want to inhibit the P140C Abel. Why? First, you can find it conserved in the evolution. It is expressed in so many cells. Even if we didn't know a lot about the function of P140C Abel at that time, it was clear that if we are making drugs to inhibit the P210 BCR Abel, we need to make sure that P140C Abel is not affected as much. So if you have a selectivity in between the oncogenic versus the normal Abel, you are likely to have more significant selectivity, not just between the P210 BCR Abel and P140C Abel, but between the P210 BCR Abel and other non-Abel tyrosine kinases. So the first go, no-go decision for this project was related to finding a way to distinguish in between the 140C Abel and P210 BCR Abel. That was the first direction that I had to take. How you can determine the specificity of an enzyme? The way to do so is based on the affinity of the enzyme with a given substrate. The substrate binding site have a certain ability to interact with the substrate. It is based on the structure of the substrate binding site. Here you have forces that are putting the substrate in place and making the substrate to bind to the enzyme. So this is one example. You have enzyme 1 and the forces are helping the substrate to bind to the active center of this enzyme. But in enzyme 2, the interaction is interfered by some repulsion forces. Why? Because the makeup of the binding site is different in between this enzyme and this one. Here you have some interferences that are making the interaction of the substrate with the enzyme not so great. When you are looking on the makeup of the substrate binding site of this enzyme, it is different as compared to the substrate binding site of the other enzyme. Affinity is the strength of interaction, the total of attractive and repulsive forces. Here, between enzyme and a substrate. If the affinity between one enzyme and a substrate is different from the affinity of another enzyme with the same substrate, then you can say that the makeup of each of the active centers is different, is distinct. That's the general idea for looking for specificity in between enzymes and their substrate. And that can be done by checking the Km value. The Km value is a measurement of the affinity between the enzyme and the substrate. The Km is the concentration of the substrate that allow 50% of the enzymes maximal activity. Low Km value means high affinity between the substrate and the enzyme and vice versa. So this is the way that you can have an indication about the structure of the active center of the enzyme and its specificity to a substrate. So my first direction in this project is to see whether or not I can find some level of selectivity in between the P140C, which you can find in normal cells, and the oncogenic versions of Abel. So I grew some cells. My incubator was full with normal cells and K562, which are CML cells with the Philadelphia chromosome, and I also grew on the side some cells which expressing the P160 gag-Abel. So my project was about the protein binding site. We didn't want to touch the ATP binding site because there is no chance to reach some selectivity at this level. But when it comes to the protein binding site, if you can show that there is sufficient selectivity in between the different enzymes, and it makes sense that it is, because they recognize different substrates. In this case, you can start to move further with the development of tyrosine kinase inhibitors to this specific site. So to make tyrosines that actually were designed to bind to the protein binding site. So that was my direction as my PhD work, to look into the protein binding site, to check if you can find some selectivity there, and then to try to fit tyrosines to the protein binding site of different enzymes. That's what we were concentrating in the lab, and that's what I supposed to concentrate in my project. But what about the ATP binding site? Although the ATP binding site was not part of my project, when I made the to-do list for the experiment to do, I put the ATP binding site as the last experiment on this list. Well, I mean, the idea is that, let's say that I can see selectivity in the protein binding site, and it would be nice to show that there is no selectivity in the ATP binding site. Wanted to do the ATP binding site as well to some level, sometimes when I will have time. And I mean, when you say that when you will have time in a very busy PhD, usually you are not getting there. But as the famous quote, men make plans and God laughs. During the time that I was working on my PhD, I had a major personal issue that I had to deal with. My younger sister, Iris, was diagnosed with osteogenic sarcoma, a very severe cancer that was found late. It was quite clear from the beginning that things are going to progress further. I decided to bring her to be hospitalized in Hadassah Hospital for two major reasons. One of them is this is the best place for treating people with osteogenic sarcoma. And second, I was there. I was 2 buildings away from the oncology department where she was hospitalized in. She had a surgical operation to remove the cancer, but she was still okay. No metastases were found up to this point. It was Wednesday. I came to the lab at 8 o'clock in the morning, and I found a note on my desk that I need to call the oncology department as soon as possible. I did it immediately, and they let me to speak with my sister. And she told me that they found something. It was clear what she's talking about. And soon enough I realized that she had some pain in the other leg, the one which is not affected by cancer, and they decided to do a simple x-ray, and they found some abnormal finding on her knee. And they wanted to start to do other imaging tests in order to see what it is all about. So Iris asked me to come to take her to those tests. She told me, when you are there, things are being done very quickly. And she was right. Because in the Hadassah Hospital, it was kind of a custom that if you're staff in the hospital, you're being treated more quickly, a bit better than others. Well, I wasn't staff in the hospital. I was PhD student in the medical school, which was affiliated with the hospital. But I had a lab coat, white lab coat. Well, can do quite a lot of miracles in this hospital at that time. So it was a tough day, a lot of roller coasters between yes, no answers. And still, by the end of the day, it was inconclusive. I took my sister back to her room. I went to the lab. I had a room for my own. I closed the door, and it was quite difficult moment. You can imagine, to understand that this is the beginning of the end. I decided that I'm not leaving the room this way, and I decided to kind of change direction by doing something in the lab. Like, to start an experiment for the next day. I had enough cells of normal Abel, I have enough cells of the oncogenic Abel, and I decided to start an experiment. Although I was quite a mess at the time, I was responsible enough to make the decision not to do a real experiment against the protein binding site that is the main direction, but to do the experiment which is the last on the to-do list, to check the specificity of the ATP binding site. Here it was not an important experiment. If it works, it works. If not, it's not. That was kind of experiment that you are doing when you are not at your best. So I started the experiment. I made extract for normal cells. I made extract from the cancer cells. And here what I need to do is to purify the enzyme out of the extract by using antibodies. So here I had two options. To mix the antibodies in the extract at room temperature for 2 hours and to keep doing the experiment until the very end. And the other option is to put the antibodies in the extract and to put the tube on ice and to put the ice in the refrigerator so the ice will not be melted overnight. So I chose the second option because I wanted to go home. It was quite late at that time. I put the antibodies in the tube with extract on ice and ran as quickly as I can to catch the last bus home. And when I was in the way, I thought to myself, did I put the ice bucket in the refrigerator? Yes, no. Well, I didn't remember. I rushed back. It was in the refrigerator. And when I came to the bus station, I realized that I just lost the last bus home. Came back to the lab, put the experiment on room temperature, and planned for processing the experiment during the entire night. At 6 o'clock in the morning, I was reading the first experiment. So I measured the KM for the ATP, for the oncogenic Abel. Well, the experiment looked nice, and the KM value for ATP on the oncogenic Abel was 0.45 micromolar. And then I read the KM experiment for ATP on the normal Abel. And here I realized that me and everybody else is totally wrong in our conceptual idea for the lack of selectivity of the ATP binding site. Because the Km value for the ATP on the normal Abel was very, very different as compared to the oncogenic Abel. Now, the kindness domain between the normal and the oncogenic, it's exactly the same sequence. However, the Km value, after all, is very different, and the makeup of each of the ATP binding site is different, is distinct. That was a very big surprise to see this. And the implication of this is that the ATP binding site can be relevant for inhibition. Brian Drucker, in his interview, was talking about the importance of this finding. when he was interviewed about the discovery of Gleevec STI-571, it was mentioned to him that it sounds like there was a catch involved. That was the interviewer question. Brian Drucker said, most people thought it would be extremely unlikely to develop specific tyrosine kinase inhibitors, particularly if you were targeting the ATP binding pocket. Researchers were thinking ATP is ATP, and 1,000 different kinases bind ATP. So you would never get specificity, but you would get a lot of toxicity. Despite this, by the late 1980s, it became clear that ATP binding site inhibitors could be developed with some specificity. As we learn more about the structure of the kinases, it became evident that there is a fair bit of variability even within the ATP binding pocket of different kinases. So specificity is in fact quite possible. When he was talking about the late 1980s, the only thing that we had that time is this. That was the information that was telling us and others that the ATP binding sites are different even if you compare them in between the different Abel proteins, oncogenic versus normal. From this point, some things were starting to take place. First, which was more relevant to my own PhD work, the chemistry portion of Alex Levitsky's lab started to make tyrosine kinase blockers that happen to inhibit the ATP binding site, usually not only the ATP binding site, but in many cases it was mixed inhibition of the ATP binding site and the tyrosine containing protein binding site. We weren't so worried to go after the ATP as well. So Israel Posner, for example, was able to show that many of the inhibitors that we have are inhibiting the ATP as well. And they can still show quite a lot of selectivity in my own experiment. So here, for example, AG957. In this experiment, I measured the IC50. The IC50 is the concentration of the inhibitor that inhibit 50% of the enzyme's activity. So the low IC50 value mean high activity of the inhibitor. The AG957 is an inhibitor with a low IC50 against the P210BCR-ABL as compared to P140C-ABL, the normal ABL protein. Again, you can see that there are quite significant differences in between the ability of this inhibitor to inhibit P210BCR-ABL very successfully and not so great to inhibit P140C-ABL. So selectivity can be achieved by using certain inhibitors, even if they are inhibiting, at least to some level, the ATP binding site. And selectivity can be achieved quite easily in between different tyrosine kinases. Here, for example, I compared several inhibitors in between P210BCR-ABL versus the EGF receptor. The data from the EGF receptor were given to me by Nir Oshrov and Pnina Yaish. They did this experiment on the EGF receptor. And what you can see is when you're using different type of inhibitors, you have different type of selectivity outcomes. The AG514 inhibit P210BCR-ABL really well, but not the EGF receptor. There are other inhibitors that are working very well, but they are not selective. AG82 can inhibit both the P210BCR-ABL and the EGF receptor more or less to the same level. And the AG537 cannot inhibit the P210BCR-ABL, but inhibit extremely well the EGF receptor. So selectivity is not a problem when it comes to tyrosine kinase inhibitors. The normal ABL proteins versus the transforming ABL proteins are different from each other. They interact selectively with ATP, with the tyrosine copolymer substrate that I'm not going to show you the data here, and the tyrosines. Selectivity can be achieved. And that was the message that we had at this stage. The information was published pretty much immediately in conferences for the formal publication, made its effect on our lab and others. After this stage, I decided to show the activity of the inhibitors in live cells. When you're moving from biochemical system to biological system, you have a lot of different things to keep in mind. Here, the drug needs to cross the membrane of the cell. And when the drug crosses the membrane of the cell, the drug may be changed by enzymes which are in the cytosol, and you can see some differences in the results when it comes to the biological experiment versus the biochemical experiment. Here I was using the K562 cells, which were CML cells. They were collected from a person with blast crisis, and they kept in culture. K562 have the Philadelphia chromosomes. They are expressing the P210 BCR-ABL. So the K562 are cells that their differentiation was arrested in the way to become red blood cells. So K562 are pre-red blood cells. They are not producing hemoglobin. They are considered to be young early cells in their differentiation. I decided to use this system in order to see whether or not the K562 can be differentiated towards red blood cells by using P210 BCR-ABL kinase inhibitors. At that time, Aviv Gazit, that was the chemist in the lab, and that's the AG, it's coming from his name, Aviv Gazit, he made a certain inhibitor. Again, the chemistry here, it's not so much relevant to my lecture, but this was an inhibitor that I was using on K562, and I was able to see some level of differentiation in those cells. Aviv took this lead and started to make derivatives to AG568. One of them was AG1112. Here you can see what he did. He kept this group, but in AG1112, he added a group which was more hydrophobic, can enter the cell probably more easily, and have some other differences. And he linked in between two groups together in order to create the AG1112. And that was the inhibitor that I found that is working at the very best. One of the ways to look for reduced tyrosine phosphorylation is by using antiphosphatyrosine antibodies. Those antibodies can bind to the tyrosine phosphorylated residues in proteins. So if the protein is phosphorylated, you're going to have a strong band, and if the protein is not, the antibodies are not going to recognize the protein. And it's not about all or none. The signals that we have from the antibodies is also correlated with the amount of the phosphorylation. I took the K562 cells, added the inhibitor AG1112, and after some period of time indicated here, I took the cells, made an extract out of them, and ran them on a gel followed by Western blot by antiphosphotyrosine antibodies. So even after 10 minutes of incubation with AG1112, you can see reduction of some of the substrates that are phosphorylated by P210 BCR-ABL. And as the time comes, you are losing the phosphorylation in more significant way. When I wanted to see whether or not the P210 BCR-ABL itself, the autophosphorylation of P210 BCR-ABL is reduced. In some cases you can see this with this exposure from here, but I decided to do a very, very short exposure to see it because the signal of the tyrosine phosphorylation was so strong, and you can see that from the beginning you have reduction of the autophosphorylation of P210 BCR-ABL. And that really tells us that the inhibitor, the AG1112, can inhibit the P210 BCR-ABL in live cells. And then I decided to look on this K562 differentiation ability after treatment with the AG1112. So the K562 are young cells that their differentiation was arrested by P210 BCR-ABL towards the red blood cells. So the idea was that if I will inhibit the P210 BCR-ABL by the AG1112, the blockage on the K562 to differentiate will be lifted, and the K562 will start to be more normal, and from our point of view is to make hemoglobin. You can see the hemoglobin under the microscope, especially if you're using a certain dye, benzidine. So it's very clear to see that the cell, the K562, are differentiated if the inhibitor is affecting them in such a way. What I saw under the microscope is the following. The blue cells are cells that are making hemoglobin. It's the certain dye that make the hemoglobin to appear as blue. As you can see here, in untreated cells, the large majority of the cells are not making hemoglobin. But if you treat those cells with AG1112, even in low concentration, which is 60 micromolar, first they are moving towards differentiation quite clearly, making a lot of hemoglobin. And there is something else that at that time we were not realizing so much. Those cells are going through apoptosis. Well, today we understand apoptosis much more than what we understood apoptosis in the early 90s. But as you can see, the cells are breaking apart. They are not spelling their guts, their cytosol to the environment, but they are turning into smaller balls which still contain the hemoglobin. Now what you say is clearly apoptosis. We didn't realize this at that time. That's actually another way to look on the inhibitors. of P to 10 BCR-ABL. If you're treating the cells with the AG1112, first, cells are going to stop to be cancer cells. They're going to move towards their normal differentiation. And as we can see it, when we started to understand apoptosis a bit better, those cells are undergoing apoptosis as well. That was the story of the early days of the tyrosine kinase inhibitors for P to 10 BCR-ABL. That was the proof of concept. Can we act against the target protein? The answer is yes, we can. The treatment can be selective enough, and the tyrosine kinase inhibitors, competitive inhibitors, are working just fine. And those inhibitors can work on the ATP binding site, can work on the tyrosine containing proteins binding site. Selectivity is not a problem. And the treatment of cells with the tyrosine kinase blockers is not toxic, but it's doing something quite specific to cells which are expressing the P to 10 BCR-ABL. The activity of the P to 10 BCR-ABL is to be stopped, and the cell can turn into other type of mechanisms that lead the cells to differentiate. So, what next? The study I conducted as part of my PhD work was completed. The articles were published. The PhD was approved. Nobody was replacing me to continue this project further in Alex Levitsky and Inon Ben-Ariah's lab. That was the last time that Alex Levitsky was working in his lab on P to 10 BCR-ABL and CML. Alex did a lot of other studies on receptor tyrosine kinases and few cytoplasmic tyrosine kinases, tried to inhibit them quite successfully with the tyrosines, but not on ABL. For several years, the field was quiet. I got my PhD, and for the next couple of years, I felt that we had an interesting story, but it seems like it is not going anywhere. But one day I was watching the program 60 Minutes, and they had a piece with a physician named Brian Drucker. And he presented there some data before publication on a successful treatment of CML patients with a unique approach. In a few minutes, it became clear that he's talking on a new tyrosine kinase blocker, and it became clear that the general approach we developed a few years earlier is working. A company named Novartis started to make more tyrosine kinase inhibitors. It's not tyrosines. They took a different type of chemical approach, and they made quite a few of those derivatives of those inhibitors, and they start to screen them against several tyrosine kinases, including P to 10 BCR-ABL. They found that several of those drugs are working on P to 10 BCR-ABL, and then they chemically developed some promisingly. They came with one or two potential drugs, made the STI 571, later on it was called Gleevec, as the drug that they were using for further analysis. This stage of the development of Gleevec was mentioned in several articles, including this one that was written by Alex Levitsky. During the early 90s, our laboratory was the first to generate selective BCR-ABL kinase inhibitors such as tyrosine AG1112 and to induce the terminal differentiation of K562 cells, originally from CML chronic myelogenous leukemia patient. And he also mentioned the AG957 I've mentioned earlier. Drucker and Lighton followed those studies utilizing the STI-571 Gleevec to produce a drug that was able to treat patients. Indeed, when Brian Drucker and others were publishing their first description of STI-571, most of the introduction was about Alex Levitsky's approach. They were talking about the Anafi et al. Studies, but about some other studies that were related to tyrosine kinase blockers, mainly on receptor tyrosine kinases. That was quite clear that the first studies about the STI-571 is an extension of other studies that were done in a small laboratory in Jerusalem. Gleevec. Well, Gleevec have several names. STI-571, the trade name is Gleevec, but scientifically it's called imatinib. So from a Brian Drucker paper where he was explaining how the STI-571 is working, so here you see the protein substrate and the ATP, which are placed in the active center of the BCR-ABL. And the gamma phosphate can be used in order to phosphorylate the substrate. So that's the normal situation where ATP can bind to the ATP binding site. But if instead of ATP you are adding the STI-571 that can bind to the very same site, ATP cannot bind there as well, and no phosphorylation is going to take place. The function of STI-571 Gleevec is through competitive inhibition of ATP binding site on BCR-ABL product. This leads to inhibition of tyrosine phosphorylation of BCR-ABL substrates. STI-571 resembles the three-dimensional structure of ATP. And by doing so, the STI-571 can enter into the ATP binding site. The STI-571 can fit perfectly into the ATP binding site and shut down the P210 BCR-ABL. In the program 60 Minutes, Brian Drucker was talking mainly about the clinical trials that he had at that time. And he was talking about phase 1 clinical trial, and this is the information that he was talking about. So phase 1 clinical trial is about the safety of a drug. That's the first stage where you're taking few people. Here you can see 83 patients with Philadelphia positive CML in the chronic phase. Here you are going to try to check not the efficacy of the drug, but if the drug is safe. That's what you want to do in phase 1 clinical trial. So the way to do this type of phase 1 clinical trial is to start with low concentration of the drug. Here it was 25 milligram per day. And then if you don't see side effect, you are increasing the dose. So patients were assigned to one of 14 doses ranging from 25 to 1000 milligram per day. And the side effect, all those concentrations, were determined as mild to moderate. And the maximal tolerated dose was not identified. In other words, even in patients that were getting the 1000 milligram per day, they had side effect, but it was tolerated. It was not side effect that stopped them from using the drug. In general, when you're doing this phase 1 experiment, you try to reach to the maximal tolerated dose in order to have a borderline that the drug is not going to be used more than a certain concentration. When you're talking about 1 gram of drug, that's a lot, and you still don't have very significant side effect at this stage. But although phase 1 is not for the efficacy of the drug, they were able to see a complete hematological response observed in 53 of 54 patients treated with 300 milligram or more per day within 4 weeks. When you check the number of cells and when you check how those cells look like, well, you don't see an abnormality after the treatment as compared to the people who are not treated. In 29 of those patients, they were able also to find major cytogenetic response. In other words, when you're looking for the Philadelphia chromosome by certain methods, you cannot find it. They were probably using the fish analysis or other type of regular methods that were used at that time to see the Philadelphia chromosome. And in 7 patients out of the 54, they had a complete cytogenetic remission, which means that even if you are doing the test in the most sensitive way that we had at the time, you cannot really see the Philadelphia chromosome in those cells. Now, it's remission, it's not a complete cure, but still, that's something that tells us that the drug is working fantastically well. They have the placebo group. When they realize that there is such a huge success of the drug, they took everybody treated below 300 milligram of the drug and the placebo group and gave all of them the 300 milligram per day, and they had quite a significant positive response for the treatment. In one of his lectures, Brian Drucker is talking about a 70 years old female as the poster patient for Gleevec. She started to enter into the accelerated phase. Her blood count started to go up quite significantly. Platelets went up quite dramatically. And he started to see blasts, which are abnormal cells. Immediately, with the treatment, the white blood cells went down to normal level, platelets went down to normal level, and blasts disappeared entirely. And that was the time that, according to Brian Drucker, he understood that, yes, the drug is working very well. So how Gleevec is working? What makes Gleevec to be such a successful drug? Gleevec is not only prevent cell growth of BCR-ABL transformed leukemia cells, but it can also induces apoptosis. In order to understand this, I want to take you back to this animation. This is CML cell with BCR-ABL. BCR-ABL is an active enzyme. This way, the BCR-ABL cannot enter the nucleus. However, when we are using the Gleevec STI 571, which can bind to the BCR-ABL, and then the BCR-ABL tyrosine kinase activity is reduced. So the cytoplasmic retention of BCR-ABL is kinase dependent. So the treatment of the BCR-ABL transformed cells with Gleevec result in the nuclear import of the BCR-ABL. Again, this is BCR-ABL, which it is inhibited from having a strong tyrosine kinase activity. BCR-ABL without tyrosine kinase activity act like a normal ABL and can move inside the nucleus. But it's not c-ABL. It's BCR-ABL with a drug which is attached to its active center. And the drug can be dissociated in some situations. That can be simply because there is a certain balance between the ABL with the drug and the ABL without the drug. It's not bound to the BCR-ABL in a very stable way. So when the Gleevec is released, the BCR-ABL is becoming an active kinase once again. Well, it's act like c-ABL when there is a DNA damage, which means that when the BCR-ABL is active, it's going to induce apoptosis. The cell can see this as the c-ABL which is induced by DNA damage. And the cell, the cancer cells, are going to kill themselves. So subsequent removal of Gleevec then result in reactivation of BCR-ABL and apoptosis. So on one hand, the Gleevec allow the BCR-ABL to enter into the nucleus, but when the BCR-ABL is in the nucleus, and since it's not going to bind to Gleevec all the time, when it is losing the interaction with Gleevec, the tyrosine kinase activity of the ABL is inducing apoptosis of the cancer cells. And that's what I can see now on the data that I had in the early 90s. We can see here quite clearly apoptosis that is expressed by fragmentation of the cells. They're keeping the cytoplasm inside them, which is part of the normal apoptosis, programmed cell death. And that's probably what we had with AG1112. The side effects which are associated with Gleevec are quite mild. Not to everybody. There are people which have side effects. But in general, when you compare the Gleevec to the other treatment that we had in the 90s and the end of the 80s, which is interferon alpha, we can say that with Gleevec we are doing far better. Patients are treated with the interferon, you can see problems in quite a few of them. The interferon is giving you a flu-like symptoms in many ways. So when you're being treated with interferon, you feel like you have a flu. And what you see here is in many ways related to the activity of the interferon directly on the immune system and other tissues. With Gleevec, we don't have the same problem. Some people are complaining about side effects from Gleevec, but it's nothing like the alternative. Most of the people who are treated with Gleevec are going to remain on the treatment for their entire life. cannot stop taking Gleevec. Gleevec is a drug that you need to take on the regular basis for your entire life in order to keep you in remission. The treatment is successful, first, because there are not a lot of side effects, and second, because we have a major progress. We have quite a significant hematological and cytogenetic responses. Almost 100% of the patients are going to have a complete hematological response for long period of time, and also complete cytogenic response. Another thing that you can see here is 5-year survival. Survival rate in patients who are treated is 95%, which is far better from what we had in the past. And the non-progression to advanced diseases, 93%. Again, I mean, it's a huge improvement. And there is another thing that you can see here. People in the United States keep coming with CML. I mean, there is nothing to stop the CML from being diagnosed. So this is quite stable. But the amount of people who are killed from the disease is going down quite dramatically. Why? Because of Gleevec. Before Gleevec, 2.4 thousand people killed from CML every year. We reached to the level of 600 killed in 2006, and it's still going down. The STI 571 have quite a high degree of specificity. STI 571 is working first on BCR-ABL. It's working on PDGF receptor, which is another receptor which appears in some cancers, and also in c-KIT. c-KIT is a tyrosine kinase receptor that can be found in gastrointestinal stromal tumor. Gastrointestinal stromal tumor that is positive for c-KIT is a disease that can be treated with imatinib, with STI 571, in order to reduce the activity of this driver mutation. So the STI 571 is working on CML, it's working on GIST, gastrointestinal stromal tumors. It has a potential to work also on PDGF receptor. But there is something else which is really cool. I mentioned to you earlier that ABL is expressed in nematodes and other worms. There are places in the world that worms infection is very common, including infection of the eye. What you see here is a certain worm which called Loa Loa. Here you can see the way that people with Loa Loa used to be treated. You can take it out from the eye by using this type of a device. You can take it out physically from the eyes, but you cannot really take the eggs away. However, scientists decided to use imatinib as eye droplets in order to kill the Loa Loa in the eyes. And it's working. Here you can see that a certain concentration, 25 micromolar and above, is killing certain type of Loa Loa. So, in other words, we are talking now about a treatment against parasites by using imatinib. I found it to be fascinating. We start with chemotherapy against cancer, and now it's time to be a treatment for a parasite which is affecting the eyes of many people in certain parts of the third world countries. But there is one thing that we need to keep it in mind. Using one drug at a time against cancer cells is not likely to work for long term. In many cases, the CML cells are not becoming resistant to Gleevec. However, there are cases that this drug suffer from the setbacks that we have for many drugs that we have around, and we are using them one by one. That's a problem here as well. And we know that there are people that after a certain period of time are going to have a drug resistant. Luckily, we have other derivatives for Gleevec that can replace the Gleevec with something which is more suitable for certain mutations. There are some specific mutations that are rejecting the STI-571. More specifically is threonine at position 315, which is turned into isoleucine in this position. Threonine can accept the STI-571, but as you can see here, the isoleucine is not. There is a certain portion of the isoleucine here which is not letting the STI-571 to fit appropriately into the ATP binding site. So that's a unique type of mutation that we were able to make a derivative for the STI-571 to fit this mutation. But other mutations are likely to take place where other drugs are going to be necessary in order to fight the mutated BCR-ABL that can reject the STI-571 or the new derivatives that we have over this molecule. I took this from Tony Hunter's lecture in 2005. What he tried to say in this lecture is that every scientist is standing on shoulders of the previous generation of scientists. CML was discovered in 1845, and in 1973 the Philadelphia chromosome was found at the first time by Janet Rowley. Then 1982, BCR-ABL was found to be a unique product which is made from the Philadelphia chromosome. And in 1984, it was found that BCR-ABL is activating cancer by elevated protein tyrosine kinase. And there is another arm of evidence. In 1970, a scientist named Abelson found the Abelson murine leukemia virus. And in 1978, we found that it is making a unique protein, which was called V-ABL, P160 gag-ABL. And later on, 1980s, ABL was found to be tyrosine kinase. But there is another arm. RSV was discovered by Peyton Rouse in 1911. In 1970s, the V-SARC was discovered. 1975, the C-SARC was cloned. In 1979, V-SARC was found to be tyrosine kinase. And according to Tony Hunter, The first protein tyrosine kinase inhibitor that were reported from Alex Levitsky labs were able to cross all the directions together. We were using all the information that we had at that time in order to come with a new field. Tyrosine kinase inhibitors as a drug for a certain cancer which is caused directly by a unique specific tyrosine kinase, BCR-ABL. And this approach was developed further towards the STI 571. Gleevec and the approach which is related to this was considered a top advances in cancer in the early 2000s. It even made the cover of Time magazine. Articles were written, Magic Cancer Bullet, Our Tiny Orange Pill is Rewriting Medical History. Books were written, and the most important things, patients are doing better.
Judy is in her 4th year of treatment for CML, chronic myeloid leukemia, a rare form of cancer. Her treatment is Gleevec, 3 pills twice a day.
Gleevec works by targeting the specific abnormality that drives the growth of one particular type of leukemia. Gleevec binds to that abnormality and completely shuts it down, so it can no longer trigger cell growth. That's very different than chemotherapy, which just non-specifically kills dividing cells.
How are you?
Brian Drucker is the lead investigator in the Gleevec study. Drucker's studies show that because Gleevec targets specific cells, it's more effective and has fewer side effects than traditional chemotherapy.
Let's have you take some nice deep breaths through your mouth, please. And once more. For patients who are diagnosed relatively early, 96%, almost all of them, have their blood counts return to normal, and typically within 4 to 6 weeks. In almost 3/4 of those patients, when you look in their bone marrows, we can no longer detect any leukemia cells unless we use extremely sensitive PCR techniques. As you know, your bone marrow looks perfectly clean.
Yes,
so
it's nice to be able to say remission.
Yes, that's right.
I mean, I didn't think I was going to say that.
While Gleevec is still a rare exception, its development demonstrates a new understanding of the diseases we call cancer.
The doctor said there was no time left and I'm still here.
Well, you're doing great. We'll get your blood counts regularly as usual.
4 years after starting Gleevec, Judy's cancer shows no signs of returning. And though this drug remains the exception for now, it's clear that our war on cancer is showing signs of success.
I'm extremely hopeful, and Gleevec has really validated decades of cancer research, and to me it's no longer a matter of can we target cancer cells. We know we can. It's just a matter of when will we get the job done.
Brian Drucker received the Lasker Award, and this is an award considered one before the big one, the Nobel Award. Many people who are getting the Lasker Award are also getting the big one as well. So this is the announcement from the Lasker Award Committee about Brian Drucker and his work on inhibitors for chronic myelogenous leukemia. And what about the study that was extended by Brian Drucker? Alex Levitsky also received another award which is also considered at the same level as the Lasker Award. Alex Levitsky had the Wolf Foundation Prize in Medicine, the Wolf Foundation Prize in Medicine. This is another a very prestigious award which considered to be one before the big one. Who knows? In the 2005 Wolf Foundation Prize in Medicine, The committee said the following. Alexander Levitsky developed specific chemical inhibitors of cancer-induced protein kinases. He demonstrated that such an inhibitor to BCR-ABL kinase induces death of chronic myelogenous leukemia cells. This is currently used with great success for therapy of patients. Alex was getting several other prestigious awards that specify the CML story, and when he's getting this award, he always communicating with me and Inon Benaria, and that what he wrote to me in 2015 after he had another prestigious award. So, dear Moti, you are co-developer with me and Inon of the foundation of Gleevec, as the 1996 Drucker paper explicitly stated. Those were pioneering days. And what about the AG1112? So the AG1112 were adopted by a biotech company named Sugen. And first of all, they started to make derivatives to AG1112. Derivatives that you can see here the general idea. I mean, you can change pretty much everything on the surface of this molecule. And they tried to do a lot of different things. But unfortunately, Sugen went down. Sugen was closed. Like many biotech companies, Sugen was not able to stay around. Alex Levitsky became a vice president in Sugen at a certain point, and he did his very best to try to move the BCR-ABL kinase inhibitors to the clinics. But the Sugen administrative refused because there are not enough patients. Well, they were really worried about the fact that they are not going to get their money back. To develop a drug or a treatment costs a lot of money. $1 billion or so. They were worried that even if they will be successful enough to get the drug, they won't be able to get their money back from the revenues that are coming from the drug. So they decided not to take this direction, and they weren't able to stay around. They were definitely wrong. When you are looking on the revenue that Novartis have from Gleevec, it's 2.5 billion in 2006, and it's going up to 3.3 billion in 2010, and it still goes up. So, Sugen did a bad decision scientifically and also economically. Novartis is getting quite a lot of money from the development of the STI 571. Alex Levitsky is my hero as a scientist. He is the father of signal transduction therapy. He did what scientists should do. Go to the scientific desert and find something new. Those are the scientific values that many of us are expecting to find around us in the scientific community. In many cases, they are there. However, in some situations, you can realize things are different. Alex is in his 80s, still very active in running his lab, and a welcomed guest in scientific conferences, especially related to cancer treatment. It was a privilege for me to work with him, and I hope you, as future scientists, will follow the values that contributed to the development of the new paradigm in the treatment of cancer. In 2011, Alex wrote me an email. I showed your pioneering work in the meeting, in the introductory lecture I had to give since Axel Ulrich, was no show. Well, a couple of days later, I wrote to him back. Hi Alex, I hope you all have a great time in the conference. I'm working on my genetic lecture about Mendel for tomorrow. However, I'm not getting very far as I keep thinking about a certain situation early 1987. Inon and I were coming in the first time to meet with you in your office. Inon, a young scientist at that time, raised the issue of publications. He was concerned that as there are too many objections to the tyrosine kinase inhibitor idea, we were likely to have significant difficulties to publish. Inon asked if there is something we can do in order to make people not to speak so loudly against the new concept. You, Alex, was wearing a very narrow lens glasses. You gave us a strong look on both of us above your glasses and said one word, שיגידו. שיגידו in Hebrew is the following: they can say whatever they want, but I will do what I think is the correct things to do. I cannot stop thinking about your שיגידו. It is coming back to me when I'm working on lectures like today on Mendel and others that change basic concept in genetics. I bet with you that at certain stage in Mendel's career, he also looked at someone directly to his eyes and said, שיגידו, in his own language. Moti. Thank you.